Okay, then chapter 8 microbes in human welfare. Let's start. Chapter 8 microbes in human welfare. Here, the topics are microbes in household products, microbes in industrial products, microbes in sewage treatment, microbes in production of biogas, microbes as biocontrol agents, microbes as biofertilizers. There are a total of 6 topics here. Let's start with the introduction. Besides macroscopic plants and animals, microbes are the major components of biological systems on this earth. We have studied the diversity of living organisms in class 11. Do you remember which kingdoms among the living organisms contain microorganisms? Monera and Protista. Which are the ones that are only microscopic? Microbes are present everywhere, cosmopolitan, in soil, water, air, inside our bodies, and that of other animals and plants. They are present even at sites they are present even at sites where no other life form could possibly exist. Sites such as deep inside the geysers, thermal vents, where the temperature may be as high as 100 degrees Celsius, deep in the soil, under the layers of snow several meters thick, and in highly acidic environments, microbes are diverse protozoa, bacteria, fungus, and microscopic animals and plant viruses, viroids, and also prions that are proteinaceous infectious agents. Some of the microbes are shown in figure 8.1 and 8.2. 8.1 we see bacteria rod shaped bacillus, here magnified 1500x, spherical shaped coccus bacteria, this is also magnified 1500x, a rod shaped bacteria showing flagella, it is magnified 50,000 times, this is flagella. Now at 8.2 viruses, a bacteriophage, this is a bacteriophage having a head, collar, tail, plate, fins and prongs. This is the caspomere, the proteinaceous shell made of caspid. B. Adenovirus, which causes respiratory infections. Rod shaped, this is rod shaped tobacco mosaic virus. magnified about 1 million to 1.5 million times. Microbes like bacteria and many fungi can be grown on nutritive media to form colonies that can be seen with the naked eye. Such cultures are useful in study of microorganisms. Colonies of bacteria showing growing in a petri dish, fungal colony growing in a petri dish. This is a fungal colony, this is bacteria. There seems to be a lag. In human health and disease, we have read that microbes cause a large number of diseases in human beings. They also cause diseases in animals and plants. But this should not make you think that all microbes are harmful. Several microbes are useful to to man in diverse ways. Some of the most important contributions of microbes to human welfare are discussed in this chapter. Microbes in the first main topic, that is, microbes in household products. You would be surprised to know that we use microbes or products derived from them every day. A common example is the production of curd from milk. Microorganisms such as lactobacillus and the others commonly called lactic acid bacteria LAB grow in milk and convert it to curd. During growth, the LAB produces acids that coagulate and partially digest the milk proteins. A small amount of curd added to the fresh milk as inoculum or starter contains millions of lactic acid bacteria, which is which in which at suitable temperatures multiply, thus converting milk to curd, which also improves its nutrition quality by increasing vitamin B12. In our stomach too, the LAB plays very beneficial role in checking disease causing microbes. This is about formation of curd. Lactic acid bacteria, the lactobacillus, are commonly called as lactic acid bacteria. They grow in the milk and converted it to curd. During the growth, a small amount of curd added to the fresh milk as inoculum or starter. This contains millions of lactic acid bacteria, which at suitable temperatures multiply, thus converting milk to curd. This also improves its nutritional quality by increasing vitamin B12 in our stomach too. The lactic acid bacteria play a very beneficial role in checking disease causing microbes. The dough, which is used for making foods such as dosa and idli, is also fermented by bacteria. The puffed up appearance of dough is due to the production of CO2 gas. Can you tell which metabolic pathway is taking place resulting in the formation of CO2? It is anaerobic respiration. Where do you think the bacteria for this fermentation come from? Similarly, the dough which is used for making bread is fermented using baker's yeast, Saccharomyces cerevisiae. A number of traditional drinks and foods are also made by fermentation by microbes. Toddy, a traditional drink of some parts of the southern India, is made by fermenting sap from palms. Microbes are also used to ferment fish, soybean, and bamboo shoots to make foods. Cheese is one of the oldest food items in which microbes were used. Different varieties of cheese are known by their characteristic texture, flavor, and taste, specifically coming from the microbes used. For examples, the large holes in Swiss cheese are due to production of a large amount of CO2 by a bacterium named Prop- Propionibacterium shermani. The Roquefort cheese are ribbed by growing a specific fungi on them, which gives them a particular flavor. Ripened by. Okay, let's read that again. We see the dough rises up in the dough batter of dosa and idli rises up. This is due to production of CO2 gas. This is released due to anaerobic respiration of bacteria. And even in case of the making bread, the dough is fermented using baker's yeast, that is Saccharomyces cerevisiae. A number of traditional drinks and foods are also made by fermentation by microbes. A traditional drink of some parts of southern India is made by fermenting sap from palms and it's called as toddy. Microbes are also used to ferment fish, soybean, and bamboo shoots to make foods, and cheese, one of the oldest food items in which microbes were used. There are different varieties of cheese depending on the bacteria microbes used. For example, the large holes in Swiss cheese are due to production of large amount of CO2 by a bacterium named uh, Propionibacterium shermani. It is Shermani species bacteria. It will release a large amount of carbon dioxide, due to which the characteristic holes we see in the Swiss cheese. And the Roquefort cheese, it is ripened by growing a specific fungi on them, which gives them a particular flavor. Now, next main topic is the microbes in human welfare. I mean, in daily house products is over. Now, microbes in industrial products. Even in industry, microbes are used to synthesize a number of products valuable to human beings. Beverages, antibiotics are some examples. Production on an industrial scale requires growing microbes in very large vessels called as fermenters. There are many uses for bacteria in the industries or microbes in the industries. Production of an industrial scale requires growing microbes in a large vessels called as fermenters. These are the fermenters here shown. Fermented beverages. Microbes, especially yeast, have been used from time immemorial, from many years, for the production of beverages like wine, beer, whiskey, brandy, or rum. For this purpose, the same yeast, Saccharomyces, Saccharomyces cerevisiae, used for bread making and commonly called brewer's yeast, is used for fermenting malted cereals and fruit juices to produce ethanol. Okay, for production of alcohols, yeast microbes have been used from many years ago. We produce beverages like wine, beer, and some of the examples given here. For this purpose, the same yeast, that is Saccharomyces cerevisiae, it is instead of that is used for bread making called baker's yeast for bread making, and brewer's yeast for alcohol production. It is used for fermenting it. This alcohol is produced by fermentation of malted cereals and fruit juices. This fermentation leads to production of less concentration ethanol. Do you recollect the metabolic reactions which result in the production of ethanol by yeast? Depending on type of raw material used for fermentation and type of processing with or without distillation, different types of alcoholic drinks are obtained. Depending on type of raw material used for fermentation and type of processing, techniques used are distillation with and without distillation. Different alcoholic drinks are obtained. Wine and beer are produced without distillation, whereas whiskey, brandy, and rum are produced by distillation of the fermented broth. By distillation may be by concentrating the drink. The photograph of a fermentation plant is shown in figure 8.5. This is a fermentation plant here. Okay, information given here is yeast is used to ferment various cereals, fruit juices, malted cereals, fruit juices to get alcohol or production of ethanol. This, if distilled or without distillation, will give different type of alcoholic drinks. Now, let's go to the next topic, that is industry under industries, that is antibiotic formation of antibiotics. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human society. Anti is a Greek word that means against the against, and bio means life. Together they mean against life in context of disease causing organism, meaning against the bacteria. Whereas with reference to human beings, they are pro-life, they are with life and not against. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes. What these antibiotics are? They are chemical substances that are produced by some other microbes and can kill or retard the growth of some specific disease causing microbes. These are substances, antibiotics produced by microbes. They kill or retard the growth of disease causing microbes. We are familiar with the commonly used antibiotic, that is penicillin. Do you know that penicillin was the first antibiotic to be discovered and it was a chance discovery? Alexander Fleming, while working on Staphylococci bacteria, once observed a mold growing in one of his unwashed culture, around which Staphylococci could not grow. He found out that it was due to the chemical produced by the mold and named it penicillin after the mold, Penicillin notatum. However, its full potential as an effective antibiotic was established much later by Ernst Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain, and Florey were awarded the Nobel Prize in 1945 for this discovery. Basically, there was a mold growing in Alexander Fleming's one of his unwashed dishes, and this Staphylococcus bacteria could not grow near that mold. And because of that, he named the fungus name was Penicillin notatum. Because of that, he named the thing as penicillin. But Much later than that, Ernst Chain, who established it as an effective antibiotic. Its actual use came after it was established as an effective antibiotic, later by Ernst Chain and Howard Florey. This antibiotic was used to treat American soldiers wounded in World War II. Fleming, Chain, and Florey were awarded the Nobel Prize in 1945 for this discovery. After penicillin, other antibiotics were also purified from other microbes. Can you name some of the other antibiotics and find out their sources? Okay, penicillin is not the only antibiotic. It is one of the antibiotics. Other antibiotics were also purified from other kind of microbes. Antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough, Kali-Kansi, diphtheria, galgotu, and leprosy, Kushi which used to kill millions all over the globe. Today, we cannot imagine a world without antibiotics. Next topic, chemicals, enzymes, and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. Examples of acid producers are Aspergillus niger, a fungus of citric acid, Acetobacter aceti, a bacterium of acetic acid, Clostridium butylicum, a bacterium of butyric acid, and Lactobacillus, a bacterium of lactic acid. These bacterium, that is Aspergillus niger, it produces citric acid, and Acetobacter aceti produces acetic acid. Butylicum, that is Clostridium, a bacterium also produces butyric acid, and Lactobacillus produces lactic acid. Yeast, Saccharomyces cerevisiae, is used for commercial production of ethanol fermentation, brewer's yeast. Microbes are also used for production of enzymes. Lipases are used in detergent formulations and are helpful in removing oily stains from the laundry. Lipases are used in detergent formulas and help in removing of oily stains from laundry. You must have noticed that bottled fruit juices brought from the market are clearer as compared to those made at home. This is because the bottled juices are clarified by the use of pectinases and proteases. They use pectinases and proteases to break down all the fruit pulp inside them, I believe. Examples are important to remember. What is the example and what it is producing. Why they are clearer than home produced fruit juices is because they are broken down. Those juices, they will be added. The juices will have proteases and pectinases added in them, so that all the plant material in them will get completely digested. That is why they look clear. Streptokinase, produced by bacterium Streptococcus and modified by genetic engineering, is used as a clot buster for removing clots from blood vessels of patients who have undergone myocardial infarction, in, infarction leading to heart attack. Okay, streptokinase is produced by a bacterium known as Streptococcus. Very, very close name, so it will help us remember. It's very important to remember. Streptokinase, it is produced by a bacterium, Streptococcus. It is modified by genetic engineering and is used as a clot buster. It is also important to remember it is used as a clot buster after genetic engineering. It is used as a clot buster and it is after genetic engineering. Another bioactive molecule, cyclosporin, cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients, is produced by the fungus Trichoderma polysporum. This is also very important to remember. Cyclosporin A used as an immunological suppressive agent during patient transplants, organ transplants. It is produced by Trichoderma polysporum. Cyclosporin A, sporin, polysporum, very close names. Cyclosporin A produced by Trichoderma polysporum helps as a immunological suppressive agent for organ transplant patients. Stains produced by the yeast Monosaccus purpureus have been commercialized as blood cholesterol lowering agent. It acts by comparative inhibiting the enzyme responsible for synthesis of cholesterol. Okay, now next one is after cyclosporin A and streptokinase. It is about stains, no, it's not stains, statins produced by yeast. Statins is a substance that is getting produced and it will reduce blood cholesterol lowering agents. They act as blood cholesterol lowering agents. They will inhibit enzyme responsible for synthesis of cholesterol, and it is produced by the fungus. No, it produced by the yeast Moniscus purpureus. There is no name relation here, but Monascus purpureus and statins, blood cholesterol lowering agents. Now let's go to next. One of the main topics here, microbes in sewage treatment. We know that large quantities of wastewater are generated every day in cities and towns. A major component of this wastewater is human excreta. This municipal wastewater is called as sewage. It contains large amounts of organic matter and microbes, many of which are pathogenic. Have you ever wondered where this huge quantity of sewage or urban wastewater is disposed of daily? This cannot be discharged into natural water bodies like rivers and streams directly. You can understand why. Because it will pollute it. Before disposal, hence, sewage is treated in sewage treatment plants to make it less polluting. Treatment of wastewater is done by the heterotrophic microbes naturally present in the sewage. This treatment is carried out in two stages. This is by heterotrophic, meaning they will use the microbes present will use the organic compounds in the sewage itself to gain energy. They will convert it into simpler compounds. And it is carried out, this treatment is carried out in two stages. Primary treatment, the treatment, this is a secondary treatment. Okay, photo of a secondary treatment. Primary treatment, these treatment steps basically involve physical removal of particles large and small from the sewage through filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration. Then the grit, soil and small pebbles are removed by sedimentation. All solids that settle from the primary sludge and the supernatant forms the effluent. Effluent from the primary settling tank is taken for secondary treatment. Secondary treatment or biological treatment. Okay, the primary treatment is about physical removal. All kinds of plastic or whatever it might be that is not organic compounds and water is physically removed. Whatever that is physically removable is physically removed. Physical removal of particles large and small from the sewage. It is done through filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration, then the grit, that is small soil and small pebbles or rocks, are removed by sedimentation. All solids that settle from the primary sludge and supernatant forms the effluent. Basically, after everything is filtered, the water that still contains many bacteria and organic compounds. The effluent from the primary settling tank is taken for secondary treatment. Secondary treatment or biological treatment. The primary effluent is passed through into a large aeration tanks where it is constantly agitated mechanically and air is pumped into it. This allows vigorous growth of useful aerobic microbes into flocks, masses of bacteria associated with fungal filaments to form mesh-like structures. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the biochemical oxygen demand of the effluent. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. Okay, let's read that again. After the primary filtration, that is physical filtration, this effluent is passed into the secondary treatment, is taken for secondary treatment. This effluent is passed into large aeration tanks where it is constantly agitated mechanically and air is pumped into it. Air is pumped into it. This allows vigorous growth of useful aerobic bacteria or microbes. And these microbes will arrange themselves in net-like structures or mesh-like structures that is called as flocks. While growing, as they are growing, the microbes will, as there is organic matter in the water or effluent, these microbes will consume oxygen that is being supplied. We are supplying air and agitating water, right? And they will consume air, that oxygen, and oxidize all the organic compounds present inside the water, thereby purifying it. And the rate of consumption of this oxygen is known as biochemical oxygen demand. Why this biochemical oxygen demand is needed for us to know, for is needed for anyone to know, is because we can identify the how much pollution the water, how much pollution the water still has. If the water is polluted still, or is it purified? To understand that, we need to know biochemical oxygen demand. If there is a high biochemical oxygen demand, that means the microbes are still purifying, still oxidizing the organic compounds in the water, in the effluent. But if there is a very less biochemical oxygen demand, it means that microbes have almost completely oxidized all of the organic compounds inside the effluent, meaning it is mostly purified. That is the reason we need a term known as BOD, the amount of, and this technically is the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. The sewage water treated till that this BOD is reduced to the minimum. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of the organic matter present in the water. The greater the BOD of the wastewater, more it is polluting. Once the BOD of the CH water is reduced significantly, the effluent is then passed into a settling tank when the bacteria flocks are allowed to sediment. This sediment is called as activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as an inoculum. Just like how we put, how we add curd to fresh milk and it will start curdling. Similar to that, how it's acting as an inoculum. Here as well, the activated sludge is full of bacteria, will act as an inoculum for the uh, aeration tank to serve as inoculum. The remaining major part of the sludge is pumped into large tanks called as anaerobic sludge digesters. Here, other kinds of bacteria which grow anaerobically digest the bacteria and fungus in the sludge. During this digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. These gases form biogas and can be used as a source of energy as it is inflammable. The effluent from the secondary treatment is generally released into natural water bodies like rivers and streams. An aerial view of such a plant is shown in the figure 8.7. Okay, what happens? We saw till the sedimentation of the flocks. After the first, let's see that treatment plan once again. First, primarily, the all the manual physical filtration, physical objects like debris, plastic, etc., will get separated through layers of filtration, sedimentation, etc. Then, activated sludge is used as an inoculum. The secondary treatment starts. Secondary, where the sludge or effluent is agitated and air is pumped, which will allow growth of aerobic bacteria. This will form mesh-like structure called as flocks, and this will oxidize all the organic compounds. This is indicated to us by BOD. When BOD becomes minimum, the effluent is taken into a sedimentation tank where all the flocks will settle, and the purified water will be pumped into natural water bodies. And after treatment about the sludge, that the flocks that got settled in a sedimentation tank, that is called as activated sludge, and this is used as an inoculum for the the secondary treatment process. And the remaining part, only small amount is required for acting as an inoculum. So remaining part of this activated sludge is taken again to be digested in, let's read that once. The remaining part of the sludge is pumped into large tank called as anaerobic sludge digesters. This is pumped into anaerobic sludge digesters. Here, other kinds of bacteria which grow anaerobically without oxygen digest all the bacteria and fungi in the sludge. During this process, gases of methane, carbon dioxide, hydrogen sulfide will be released. And this will act, because methane is inflammable, it will act as an energy source. The plant is shown here, an aerial view of a sewage treatment plant. You can appreciate how microbes play a major role in treating millions of gallons of wastewater every day across the globe. This methodology has been practiced for more than a century now. In almost all parts of the world, till date, no man made technology has been able to rival the microbial treatment of sewage. You are aware that due to increasing urbanization, sewage is being produced in much larger quantities than ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities, so the untreated sewage is often discharged directly into rivers, leading to their pollutions and increase in waterborne diseases. The Ministry of Environment and Forests has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged into the rivers. A visit to sewage treatment plant situated in any area near you would be very interesting and educating experience. Now let's go to the next main topic, microbes in the production of biogas. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity which may be used as a fuel. You have learned that microbes produce different types of gaseous end products during growth and metabolism. The type of gas produced depends upon the microbes and the organic substrates they utilize. In the examples cited in relation to fermentation of dough, cheese making and production of beverages, the main gas produced was carbon dioxide. However, certain bacteria which grow anaerobically on cellulosic material produce large amounts of methane along with carbon dioxide and hydrogen. Certain bacteria grow anaerobically on cellulosic material. Cellulose is basically plant material. These bacteria are collectively called as methanogens. And one such common bacterium is methanobacterium. These bacteria are commonly found in the anaerobic sludge during sewage treatment. These bacteria are also present in the rumen, a part of stomach of cattle. We have studied about this in biological classification about archaebacteria, strong bacteria that can survive in even ruminant parts of the cattle's gut of the cattle. They are called as methanogens. They are methane producing organism. One of the bacteria is called as methanobacterium. A lot of cellulosic material present in the food of cattle is also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of the cattle. Do you think we humans are able to digest the cellulose present in our foods? Thus, the excreted dung of cattle, commonly called as gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called as gobar gas. The biogas plant consists of a concrete tank 10 to 15 feet deep, in which bio-wastes are collected and a slurry of dung is fed. A floating cover is placed over the slurry, which keeps rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. The spent slurry is removed through another outlet and may be used as fertilizer. Cattle dung is available in large quantities in rural areas where cattle are used for variety of purposes. So biogas plants are more often built in rural areas. The biogas thus produced is used for cooking and lighting. The picture of a biogas plant is shown in figure 8.8. The technology of biogas production was developed in India mainly due to efforts of Indian Agricultural Research Institute IARI and Khadi and Village Industries Commission KVIC. It's important to remember these names: Agriculture Research Institution and Khadi and Village Industries Commission. If your school is situated in a village or near a village, It will be very interesting to inquire if there are any biogas plants nearby. This is the biogas plant and learn more about it from the people who are actually managing it. This is a diagram of a typical biogas plant. This is a gas outlet here. There is a floating cover which will rise as the gas production increases. This is the digester. This is dung water here. Insert and sludge is removed here. Gas outlet CH3 and some other gases are produced, which is removed through outlet here. Now let's go to the next main topic, that is microbes as biocontrol agents. Only 2 pages. Biocontrol refers to the use of biological methods for controlling plants, diseases, and pests. In modern society, these problems have been tackled increasingly by the use of chemicals, by use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike and have been polluting our environment, soil, groundwater, fruits, vegetables, and crop plants. Our soil is also polluted through our use of weedicides to remove weeds. Biological control of pests and diseases in agriculture. There is a method of controlling pests that relies on natural predation rather than introduced chemicals. In agriculture, method of controlling pests that relies on natural predation rather than introduced chemicals. A key belief of the organic farmer is that biodiversity furthers health. The more variety of landscape has, the more sustainable it is. The organic farmer therefore works to create a system where the insects that are sometimes called pests are not eradicated but instead are kept at manageable levels by a complex system of checks and balances within a living and vibrant ecosystem. Contrary to the conventional farmer practices, which often use chemical methods to kill both useful and harmful life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the webs of interaction between the myriad of organisms that constitute the field fauna and flora. The organic farmer holds the view that the eradication of the creatures that are often described as pests is not only possible but also undesirable, for without them the beneficial predatory and parasitic insects which depend upon them as food or hosts would not be able to survive. Thus, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as pests, and also their life cycles, patterns of feeding, and the habits they prefer. This will develop appropriate means of biocontrol, basically biodiversity. Biodiversity is better than using chemicals. The very familiar beetle with red and black markings, the ladybird, and dragonflies are used to get rid of aphids and mosquitoes. Important to remember. Ladybird and the dragonflies are used to get rid of aphids and mosquitoes, respectively. Ladybirds get rid of aphids and dragonflies get rid of mosquitoes. An example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis, often written as BT. These are available in sachets as dried spores, which are mixed in water sprayed onto vulnerable plants such as brassicas and fruit trees. When they are eaten by the insect larva, the gut of the larva, the toxin is released and the larva get killed. That is because the Bacillus thuringiensis bacteria release during a development stage of their life release a crystal toxin which is inactive in normal state but gets activated in the insect gut to kill them, to attack their midgut epithelial cells, and by swelling of the cells, stomach cells, it will kill the bacteria. The bacterial disease will kill the caterpillars but leave other insects unharmed. Because of the development methods of genetic engineering, in the last decade or so, the scientists have introduced B-thoragenesis toxin genes into plants. Such plants are resistant to attack by black pests, insect pests, which we saw in biotechnology. BT cotton is one such example which is being cultivated in some states of our country. You will learn more about this in chapter 10. Biological control being developed for use in treatment of plant disease is the fungus Trichoderma. Trichoderma species are free-living fungi that are very common in the root ecosystem. They are effective biocontrol agents of plant pathogens. Okay, what is a biological control being developed in treatment of plant disease is the fungus Trichoderma. Trichoderma species, they are free-living fungi, they are not parasitic. They are very common in root ecosystems. They are effective biocontrol agents of several plant pathogens. They act as biocontrol agents for several plant pathogens. Baculovirus are pathogens that attack insects and other arthropods. The majority of baculoviruses used as biological control agents are in the genus Nucleopolyhedrovirus. These viruses are excellent candidates for species-specific narrow-spectrum insecticidal applications. They have been shown to have no negative impact on plants, mammals, birds, fish, or even non-large insects. This is especially desirable when beneficial insects are being conserved to aid in all overall integrated pest management (IPM) program, or when an ecologically sensitive area is being treated. Okay, these baculoviruses are pathogens that attack insects and other arthropods, and majority of these baculoviruses are used as biological control agents and are in the genus Nucleopolyhedrovirus. These viruses are excellent candidates for specific narrow spectrum insecticidal application, meaning they will kill only specific desired insects. I mean undesired insects. Only targeted insects they will kill. They have shown no negative impact on plants, mammals, birds, fish, and untargeted or yes, on non-targeted insects. This is especially desirable when beneficial insects are being conserved to aid in overall integrated pest management program, or when an ecologically sensitive area is being treated. Non-targeted insects, they can be aphids, they can be not aphids, ladybird and dragonflies, where ladybird hunted or killed aphids which were dangerous, and dragonflies controlled mosquitoes. Because of that, we don't want to kill those insects. They are beneficial. Microbes, as now next main topic is microbes as biofertilizers. With our present day lifestyle, environment pollution is a major cause of concern. The use of the chemical fertilizers to meet the ever increasing demand of agriculture produce has contributed significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of chemical fertilizers, and there is a large pressure to switch to organic farming, the use of biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of the biofertilizers are bacteria, fungi, and cyanobacteria. You have studied about the nodules of the root of leguminous plants formed by the symbiotic association of rhizobium, leguminous, leguminous family specialty. They fix nitrogen, they get food from plant, they provide nitrogen to the plant. These bacteria fix atmospheric nitrogen into organic forms, which is used by plants as nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, okay, these bacteria, that is rhizobium bacteria, fix atmospheric nitrogen into organic forms, which is used in plant nutrition. Other bacteria can also, other free living bacteria, they're not associated with plants. These also live in the soil, can fix atmospheric nitrogen while free living in the soil. Examples for those are azospirillum and azobacter, thus enriching the nitrogen content of the soil. Fungi are also known to their symbiotic association with plants. Mycorrhiza, many members of the genus Glomus form mycorrhiza, the fungal symbiont. In these associations, absorb phosphorus from soil and pass it to the plant. Use of this is mycorrhiza, association between fungi and the roots of higher plants, is that this fungus absorbs phosphorus from the deep soil and gives it to the plant. Plants having such associations show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in plant growth and development. Can you tell what advantage the fungi derives from this association? It derives food. Plant will provide food. Fungi will provide protection from root pathogens, absorb nutrients from the soil, that is phosphorus mentioned here. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments. Cyanobacteria are blue-green algae, which are photosynthetic, can fix atmospheric nitrogen, like nostoc, of which can fix nitrogen. Example, Anabina, nostoc, oscillatoria, etc. In paddy fields, cyanobacteria serves as important biofertilizers. Blue-green algae also add organic matter into the soil and increase its fertility. Currently in our country, a number of biofertilizers are available commercially in the market. These regularly in their fields to replenish the soil nutrients and to reduce dependence on chemical fertilizers. Summary: microbes are a very important component of life on Earth. Not all microbes are pathogenic. Many microbes are very useful to human beings. We use microbes and microbially derived products almost every day. Bacteria called lactic acid bacteria grow in milk to convert it into curd. The dough, which is used to make bread, is fermented by yeast called Saccharomyces cerevisiae. Certain dishes such as idli and dosa are made from dough fermented by microbes. Bacteria and fungi are used to impart particular texture, taste, flavor to cheese. Microbes are used to produce industrial products like acetic acid, alcohol, lactic acid by lactobacillus aceti is a species name for the bacteria producing acetic acid, which are used in a variety of processes. In industry, antibiotics, penicillins produced by useful microbes are used to kill disease-causing harmful microbes. Antibiotics have played a major role in controlling infectious diseases like diphtheria, whooping cough, and pneumonia. For more than 100 years, microbes are being used to treat sewage, by the process of activated sludge formation, and this helps in recycling of the water in nature. Methanogens produce methane biogas by degrading plant waste, biogas produced by microbes, which is a source of energy in rural areas. Microbes can also be used to kill harmful pests, a process called as biocontrol. The biocontrol measure helps us to avoid heavy use of toxic pesticides. For controlling pests, there is a need these days to push for use of biofertilizers in place of chemical fertilizers. It's clear from the diverse uses human beings have put microbes to that they play an important role in the welfare of human society. So final conclusion is that they play an important role in the welfare of human society. So that is with the topic name, that is microbes in human welfare, starting and ending with the topic name. Okay, then first revision completed.